Okay, good afternoon, everyone. We're going to go ahead and get started since we're at 1:30. My name is Candace Hart. I'm the State Master Gardener Specialist here for University of Illinois Extension. And I'm just on today to help organize this meeting and welcome everyone. But our researchers for the I-Pollinate project are going to be doing the presentation today. So we'll have time at the end for questions, I'm sure, and you can type those into the chat box as we're going along. I've muted the whole audience, so if you have questions, you can type them into that chat box, and we will address those when we get towards the end of the presentation. So Morgan, I'm going to hand it off to you, and then we can have introductions of everybody else who's on today.
Awesome. Thank you so much, Candace. Can everyone hear me okay?
Yep, I can.
Wonderful. Okay. As Candace mentioned, my name is Morgan Mackert. I'll introduce myself in just a few minutes, but I just wanted to say a quick thanks for taking the time out of your day to come to this webinar. We really appreciate it. As Candace mentioned, this webinar is for I-Pollinate, a citizen science organization project that we put together here at the University of Illinois to gather information and data about pollinators in the area. Without further ado, let's get started. So a quick background about the scientist team in the I-Pollinate project, starting with Dr. David Zaya there on the left. Dr. Zaya is a member of the Illinois Natural History Survey, and he heads the Plant Pollinator Ecology Program. Specifically for I-Pollinate, he heads the Monarch portion. Again, we'll talk about that in just a few minutes. Michael McKelvey is part of the Office for Mathematics, Science, and Technology Education, and he leads the BeeSpotter portion of I-Pollinate. And Dr. Alexandra Harmon-Threet, the third picture there, originally designed the CU Pollinator Count program, and she is a member of the entomology department as a faculty member. And then there on the far right, you will see yours truly, Morgan Mackert. I am a member of the Harmon-Threet Lab here at the university, and I am sort of the contact person for I-Pollinate. So if you have any questions whatsoever, please feel free to reach out to me. Next, we have our lovely extension team, starting with Deborah Seiler, who is a member of the Extension and Public Engagement Program. She's their coordinator. We have Kelly Alsop, who is an extension educator in horticulture. Our very own Candace Hart, the State Master Gardener Specialist, and Christopher Evans, who is a Forestry and Extension Research Specialist. Okay, now that we're familiar with all the names and faces behind the scenes, let's start with a little background about I-Pollinate, which originally started due to a dramatic increase in public interest in pollinator conservation as well as citizen science. So the university had 3 independent programs, the first of which the Plant Pollinator Ecology Program at the Illinois Natural History Survey. As I mentioned, Dr. David Zaya is a part of that. Secondly, BeeSpotter, which Michael McKelvey is leading. And thirdly, the CU Pollinator Count, which Dr. Alexandra Harmon-Threet designed. So the university, the extension office approached these 3 independent programs with the intention of bringing them together due to all of these programs having the same goal of increasing data collection on native pollinators with the intention of improving conservation efforts. They all have the same goal, so why not bring them all under the same sort of umbrella program in an effort to provide the public with a single centralized location to learn about as well as participate in citizen science projects that would benefit our pollinators. So moving on, ultimately, the goals of I-Pollinate are, number one, to report bumblebee sightings to BeeSpotter. And BeeSpotter looks a little something like this. You can go ahead and register at the BeeSpotter website. You can take photos of any bumblebees you see. Those photos will then be identified by an expert, and we will have then a record of that bee being in that area that you found. And that will provide information on the demographics as well as the ranges of different bee species in the area. Secondly, we wanted to explore monarch responses to milkweed implementation and garden variability. And lastly, we wanted to determine ornamental flower attractiveness to pollinators. And for our purposes, pollinators are anything that visits a flower. Bees, flies, butterflies. Ants can be considered pollinators, all sorts of insects that visit flowers and have the potential to transfer pollen. So now that we know the goals, the way that we are collecting these data are by establishing a research garden. And this research garden is composed of 6 plant types of your choosing from the 12 listed on the right side, excluding the swamp milkweed. So we've got 12 options that you can choose from. You choose 6. You go ahead and plant those in any configuration that seems feasible. The photo here is simply an example. You'll notice that the M's indicate swamp milkweed. That milkweed is planted for the monarch caterpillars and butterflies. However, if you're not monitoring monarchs, you can go ahead and substitute another plant type instead of the milkweed. And we do have I-Pollinate signs coming soon. So you can go ahead and pop a sign next to your research garden, get the word out about I-Pollinate, and let people know that you are a citizen scientist conducting important research. Now, not an issue, but something that we ran into last year in 2019, where maybe you don't have a lot of space, or maybe your garden is full of flowers and you don't necessarily have room, and you don't want to dig up a current flower bed or what have you. New this year, we are offering an alternative to planting your research garden in soil in the ground. You can feel free to use containers. Plant your garden in containers. The data will be collected in the exact same way. We simply ask that you indicate that you have a container garden so we're aware of that. Now, when you are looking to establish your garden, we have a buying guide. The I-Pollinate buying guide is available online. Here's the website for I-Pollinate. You'll simply go to the buying guide. It looks just like this here on the right. It's 3 pages long, and it offers descriptions of every plant that we would like you to purchase and establish in your research garden. Again, you don't have to purchase all of them. Simply choose 6 or 7 of your choosing, the ones that are available, colors that you like, pricing, whatever decision that you make that determines which flowers you choose is just fine, so long as they're ones that are on this buying guide. Okay, so now we've got our garden designed, we've got it planted, it's established. The next thing we want to do is start collecting our data. Data collection weeks are the 3rd full week of each month from June through September. Now, in the current climate of the world, considering the virus that's going around, we are not encouraging you necessarily to establish your garden, to plant your garden in June, simply because we can't encourage you to go purchase these plants from the public. So, little asterisk there by June. If you've already established your garden, that's wonderful. We will absolutely collect those data. However, for the majority of people, we are expecting data collection to start perhaps in July, fingers crossed. Regardless, data collection will continue through September. One quick caveat though, we do ask that data collection occurs in that third full week of each month. But if you are so inclined, we will absolutely accept any other data. So if you want to collect in the first and third full weeks of the month, or if you get a wild hair and you just want to go collect data every single week, by all means, feel free. That would absolutely help us out a ton as well in terms of weather anomalies. So any data that you feel inclined to submit, we will gladly accept it. Now, in terms of data collection, this is what the floral survey data sheet will look like. We start by asking simply to estimate the overall health of your garden on a scale of 1 to 5. Are the plants living or dead? Do they look damaged? Is the color a little bit off? Anything like that. Next, we're interested in determining the context of your personal yard and any gardens that you have. So we ask 2 questions here. First, approximately how far away are other flowering plants from the edge of your I-Pollinate garden? And secondly, we're interested in other plants that you have in your garden that are not part of your I-Pollinate research garden, but other plants that are seemingly attractive to pollinators. And the reason that we ask this is because it's important for us to know the context of your yard and garden, because perhaps, maybe you establish your research garden in the example photo here on the left. Those flowers might be acting as a beacon and attracting lots of different pollinators from all over, because there really isn't anything else for the pollinators to visit. However, if you would establish your research garden in the example on the right, it might get lost in all of the different flowers that are available in that very diverse, very abundant garden. So that's why we're curious about the context of your personal yard. Secondly, we're also interested in the diversity of your neighborhood. Again, do you live in an area where all of your neighbors have super diverse, very abundant gardens, or maybe not so much? Again, just for example, if your neighborhood looks like on the left, looks like the neighborhood on the left, and you establish a garden, it might, that context might influence the pollinator visitation rates versus if you establish a research garden in the example on the right. So that's all of the floral survey. Secondly, we have the Monarch survey. Simply what you'll do is look at each living milkweed plant and look at all of the leaves, see if there are any monarch eggs and or larvae there. If so, we just ask that you record how many, as well as any dead or damaged eggs or larvae. And lastly, we have the Pollinator Survey. In the Pollinator Survey, all you do is for each type of annual ornamental, you'll select 2 plants of the 4 that are established, and you'll watch the flowers on those plants for just 3 minutes. If a fly lands on one of the flowers of the 2 plants that you're watching, you simply make a tally mark in the fly box next to in the ornamental column that you are recording. If nothing visits the flowers, you simply record a 0. And I do want to emphasize that if no insects visit the flowers, record a 0, and please do enter those data because it is very important for us. For example, say you go to a reconstructed prairie and you visit that reconstructed prairie several times over the growing season, and you never see a single bee or never see a single butterfly. That's really important for us to know, because there's something going on there that that those plants are not supporting pollinators. Something is amiss there. So zeros are still data. They are very important, and we sure would appreciate those getting submitted as well. As far as submitting data, you'll just go to the I-Pollinate website, which is here. You'll click on the I-Pollinate Research Garden dropdown and click on step 4, where it says record and report data. That will take you to a Google survey, just like this one. This one is for the Floral Survey, and it'll simply ask you to input all of the information that you recorded on the data sheets, or you have the option to directly input your data into the Google survey from your smartphone, from an iPad, whatever works for you. Next, I wanted to share some results from I-Pollinate 2019, our pilot year. We were very pleased with the results that we received. A total of 125 floral surveys were conducted, which is astronomical. Considering the scientist team and the extension team, there's no way that we could go out and collect all of this data. So we are very, very appreciative of the dedication and time that our volunteers and participants put in. One thing that we did notice, though, was that the number of submissions declined over time. So we are brainstorming ways to keep participation high throughout the entire year. Next, we observed a big diversity of flower varieties blooming in people's yards. As you can see, there were anywhere from one blooming flower variety all the way up to over 70 different types of flowers in people's gardens. So that is a great span of different garden diversities, which is going to offer a lot of insight. Secondly, for the Monarch surveys, we had, again, a total of 130 Monarch surveys submitted. Again, though, we do see that same decline in submissions throughout the year. And for some results of the Monarch survey, you'll see here on the box in the right, the yellow box, the way that this is presented, we have the range. So for instance, eggs 0 to 11. So one person, several people submitted 0 eggs, all the way up to someone observing 11 eggs on one plant. The percentage in the parentheses is the percentage of zeros that we received. So while we do have a lot of zeros, as I mentioned, that is very important for us to know. We did see some pretty large numbers of monarch eggs and larvae using these milkweeds. Very important information. Moving on to the pollinator surveys, we got a total of 744 pollinator surveys submitted, which is just phenomenal. However, we do see that same decline in submissions throughout the year, so we're going to hope to keep motivation high throughout this 2020 data collection year. As far as the total number of plants visited by pollinators, total number of plants observed, we see that pollinators visited marigolds, geraniums, and lantana pretty frequently. However, looking at the data another way, by looking at the average number of insects observed on each plant, we see that Cleum, Euphorbia, and Scaevola are also very attractive. So we're hoping to sort of tease apart which plants are most attractive to pollinators using the data collected from this year. The number of pollinators observed per sample, again, the data is presented in the same way as the monarch larvae and eggs. A lot of zeros for the bees and butterflies, less so for the flies, but we observed up to 20 bees per sample visiting one plant type, 11 butterflies and or moths, and 21 flies. Very cool. And then some results for BeeSpotter. So averaging the past 2 years of BeeSpotter data, after I-Pollinate originated, we saw a 46% increase in user signups. We saw a 30% increase in the number of users submitting a spotting. And lastly, we saw a 22% increase in total spottings submitted. Now, for our second year of I-Pollinate, we are hoping for some goals, and we've got some improvements in mind. Now, the goals for this year are to increase participation. We want more participants throughout the state so we can get an even better view of what the pollinators' demographics and the ranges are throughout the state. We're hoping to increase the confidence of participants in their data collection by hosting trainings, by hosting webinars such as this. And thirdly, we wanted to streamline data submission. During the last year, we conducted a survey of the participants and asked what we could improve for them. And here, number 2 and 3 here of the goals were ones that were requested by the participants. So we've been working very hard to accomplish those improvements. And now for 2020, just a reminder, these are the weeks that we are hoping to collect data. Again, the 3rd full week in each month from June, tentatively, July, August, and then on through September. Here again is the I-Pollinate website. It's got all of this information that I've presented plus a ton more. We've got identification resources there. We've got other webinars there. So wealth of information on the website. You can also, if a question pops into your head that you didn't have a chance to ask during this webinar, go ahead. There's a place that you can submit questions on the website, and they will come straight to me and I will get right back to you. And with that, I'd like to open the floor to any of the other scientist team or the extension team to clarify things that I might have misspoke about or provide additional details or emphasis on certain points.
It looks like Alex has been answering a lot of the questions in chat. Kelly, do you want to maybe point out some that have been missed maybe?
Catherine asks, why did you choose non-native species for the ornamental plant portion of the project?
Yeah, I guess I'll answer that. If you guys have participated in the past, you probably heard me say that, so this is Dr. Harmon Reed, and I'm interested, one of my research questions is about ornamental plants. I'm a big gardener. I love to garden. I love pollinators too, because I'm a pollination ecologist. And one of the challenges that I find in my garden, I live on the corner, and I can't put a lot of native plants in my front yard because most of them they grow too tall. And I get cited by the city every year for my native plants that are too tall. But there are lots of other plants, ornamental perennials, ornamental annuals, that probably provide some kind of resources, but there's very, very little data on them. And so I saw this as an opportunity to start bringing in some of that data both by understanding what's going on in people's yards in general in terms of floral diversity, and then also how those annual ornamental plants are performing. Now, the list that we've given you is a list of plants that are either that we don't have very much data on, or are very commonly suggested as being quote unquote good for pollinators. But the data is actually pretty thin. And so this was an opportunity to get a lot more data, to offer people some alternatives if they're in a situation like I am, and they can't put in a lot of native plants in various parts of their yard, or for neatness laws, if you live in particular for HOA requirements, or if you are a temporary home, where you're living is only temporary, you're not necessarily homeowner, but you want to do something, this would open up a lot of opportunities for other people.
So Alexandra, go ahead and answer this question. Patty asked about the containers. Do you want them configured like the beds? And are there any other requirements for putting an I-Pollinate garden in containers?
Okay, this is a great question, and this is a new thing for us, the containers. So we don't really have a lot of recommendations for like how big the containers should be. Ideally, yes, if they are in the same configuration, that obviously helps us. One of the big challenges for us as scientists is reducing data variability or understanding data variability. And so having them in a similar configuration really helps maintain continuity between the data that we get, if that makes sense. But if you find yourself only able to put out a group of 4 geraniums, go for it. I still want to encourage people to do that. And because when you submit a survey of like what plants you put into your I-Pollinate garden, and also what what else is blooming in your yard, so we will get that kind of like back data to say that you didn't end up putting in all of the plants. Because we also anticipate like some of them might die and or not be flowering at various points throughout the summer. So we do get that information. So if you, ideally, yes. If you can't get there, that's okay too. We have back ways of controlling for it on the back end with the data.
Back to you again. Patty asked, do you want them to do observations the same time each day? And then, also address when I was doing my stuff last year, if it was a windy day, did you want me to come back on a non-windy day? Because I definitely noticed I didn't get very many observations on windy days.
Okay, so trying to make sure I'm holding all those questions in my mind. You don't necessarily need to do the surveys at the same time of day. Any time when the sun is shining is great. I would avoid the hottest part of the day, both because it's uncomfortable for you, but then also pollinators, insect pollinators in a way, tend to have much lower activity during that like really peak hot time of the day between 12 and 3. So if you do them early or late, that's fine. Early you tend to see a little bit more. If it's windy, that is challenging. We do see a lot of pollinators that will move around in the wind, but you're definitely going to see fewer things. So this is one of the reasons that we try and have people collect as much data as they can during the same week. Someone did ask if they needed to sample the entire week, and the answer is no. We're hoping for each plant species, you can do 3 to 4 observations on each of them, or more. Obviously, if you can do more, that's great, but a few observations on each plant species in your yard across that week would be great. And we know that some days it's gonna rain, which is why we left a whole week open, right? Maybe you have to work some days and you're not able to do it. So the week was just to give people a little bit of flexibility, but also to allow us to make sure the data was kind of coming in chunks rather than really kind of spread out over the entire month or summer, making it much harder for us to make comparisons between, a yard in the northwest suburbs of Chicago and in a county in the southern part of the state. So we were trying to be able to make those meaningful comparisons. So which is why we're trying to limit the times when people are doing sampling. But again, if you want to collect data all of the time, go for it. My dream is that someone, when they're having their cup of coffee, can just like wander out into their garden and like do a 3-minute observation, right? They're only 3 minutes. So you could have a sip of coffee, observe your plants for a few minutes, and then, you know, upload that information for us very casually. So it becomes like a kind of normal part of your day, which is what I would dream of. Like, that's how I dream. I was like, I would love to just be able to wander into my yard and collect data and like feel like I'm, you know, starting my day very productively.
Thank you. Morgan, can you describe the registering process, and then so when they register, they're registered, and then they just come back during the time periods and submit their data?
Absolutely. So I'm just going to zoom back here. Sorry for any motion sickness that this imparts. So here I've got the I-Pollinate website. This is what it looks like. Again, here's the link if you need it. We've got on the left-hand side there in the blue, you can see where it says information sign up. If you need more information, go ahead and input your information, your contact information there. However, I want to direct you to the I-Pollinate Research Garden heading. The drop-down list under there, step number 2 is where you're going to register your garden. While you can still submit data without registering, we would really request that you would register your garden, simply because registering your garden gives us sort of just one list where we have everyone's contact information, such as the location of the garden, your email address, your name. So if we find an anomaly in the data and we need and we would like to contact you about it, we can simply go to that registration list, find you, and be able to contact you quickly. Last year, we also sent out sort of a preliminary results newsletter with a little thank you in the form of milkweed seeds. So, you know, just for participating with us and helping us collect all of this data. So if we are, I hope to do it, if we're going to do it again this year, that offers us a really clear way to see who is participating and have their contact information compiled succinctly.
Okay, I guess this could be for David and Alexandra. Janet asks, what if one of the plants dies? Do you want them to replace them?
Okay, I'll speak. No, I think actually it probably will be really difficult to replace them. And if it happens early on, you know, when you first put your garden in, I actually was fortunate enough to already get all of my plants, and my garden is already in, although it's supposed to frost here tomorrow night, so we'll see what happens with that. I'll probably try and put a blanket around it. But you know, if I lose some of those in the next few weeks and I feel like I can replace them, please go ahead. But as we move further into the summer, it will be really difficult to do so. So don't feel like you need to. And I do think that there is, in all of, I know it feels like a lot of sheets, but for us, this stuff was really important, because if you lose a plant, we actually have a mechanism for you when you talk about the health of your plants, you actually can tell us that one of them died. So I know it seems like a lot of sheets, but all of this kind of helps us better understand, how does your I-Pollinate garden look? How does your whole, how does the rest of your garden look? And things that, and we do ask for some additional pieces of information, like how often do you water your plants and things like that? How much mulch do you use? All of these things are actually really important to insects. And we thought it was important for us to try and record as many of them as we could. I know it feels a little overwhelming, but I promise you there's always like a method behind our madness of why we're trying to ask some of these questions. So hopefully that comes through this presentation, and I'm happy to answer any other questions that you have. I've been trying to ferociously typing, trying to get to all of the questions. And I know that, I don't know if David's on today, but we definitely had some mortality with the Asclepias plants last year, the milkweed plants, because they just seem to get decimated by aphids. Like mine looked absolutely horrendous. The rest of my garden looked really good, and my milkweeds were just little sticks, leafless sticks, because the aphids just really did a number on them. So I just reported that as part of my data collection. They did really badly. So, you know, there wasn't going to be any way for me to replace them, but I think that that information was still very valuable.
So I'm going to just really quickly address Michelle's question. The garden diagram is on the I-Pollinate website, along with other webinars that the scientists did last year, which can be extremely helpful if you guys want to go to the website. The area of I-Pollinate is the state of Illinois. If you live in Illinois, you want to do an I-Pollinate garden, that's wonderful. The other one is, can I plant near a native pollinator garden? And absolutely, what you're going to actually record that information when you type in your floral survey. And so, you know, what I did last year is I looked around and I was like, oh, there's a flower, there's potatoes flowering within 15 feet of the garden. And then it asked me to estimate the number of flowers. And of course, I didn't go through and count every potato flower. I just said hundreds. And so you can plant it near a garden. You just need to make sure you tell them that in your floral survey. And then Catherine asked about distinguishing between the 5 instars. And Alexandra put up a guide that David had. There's tons of information out there on this, the Monarch joint venture. There's great YouTube videos out there on how to tell the different instars. It's usually variation between the tentacles, the size of the tentacles, the size of the caterpillar, and the striping. After you get it down, it was actually kind of easy for me to be able to distinguish the instars. And David said, if you record a second instar and it's really a first instar, you're not really going to mess up his data that much. So don't feel discouraged about identifying those instars. Now, Carla asked, are these the only plants available to use in the I-Pollinate garden? And are we selling them, or how are they supposed to access these? I guess I could answer that question too. So yes, Dr. Harmon-Threet is specifically looking for information on these 12 ornamental plants, because she does want to be able to give recommendations to people like us in the future. Hey, if you plant Cleome, you'll be contributing to pollinator health. So these specific plants are what we want to use. And are we selling them? No, unfortunately not. We don't have that kind of system set in place. You can go to your local garden center following all the guidelines and purchase these plants. We cannot do an I-Pollinate garden for Master Gardener projects until July 6th. So we're encouraging you to do these in your home gardens. And Chris and Candace are going to give you volunteer credits for doing this project. And then, oh, this one's a good one for Alexandra. How do you know for sure if you're getting organic versus treated plants in the industry? And is that something that they have to make sure that they got organic plants?
That's a great question. And this was one of my big concerns about this project. We don't control for that right now. We do discourage you from treating for things once they're in your yard, but there's basically no way for us to control it because we have such a wide distribution across the whole state and we can't control the supply of the plants for everyone. We just that's not something we're able to control right now. Fortunately, I was reading something the other day that they had discouraged the use specifically of things like neonicotinoids in ornamental plantings. So I don't think that most of the plants are treated anymore. If someone knows better, please let me know. I think I read that somewhere that that was no longer that was one of the recommendations, that they stopped treating these ornamental plants. Before, I have bought plants previously, ornamentals, that they specifically had little tags on them that said they had been treated with neonics, but that was a number of years ago, and now they seem to have stopped doing that. So that's good for us. That doesn't mean they're not sprayed with other things, but again, because we have such a wide distribution across the state, we just can't control for it.
Alexander, can they start these plants from seeds, and can they choose any variety they want?
Yep, varieties. In the planting guide, there are some listed varieties, but just go with color and the type as best you can. Unfortunately, there's so many varietals of many of these, especially things like lantanas. Even like a yellow lantana, there's probably a few dozen varieties of that. We can only do what we can do. And I was sharing in the chat that we recently got some funding to try and help supply some of these plants. So maybe in the future, we'll be able to control a little bit more of those pieces. But right now, don't worry about it. Just try and go with the color and species that we're hoping for. And I'm sorry, there was a second part of that question. Oh, seeds, yes. You can, if you think you can get them flowering before we want data to start being collected in June, or even July, that's totally fine. I definitely planted some of my alyssum as seeds because I just was having a really hard time finding it, and it flowers really quickly, so I didn't have a problem getting it up to flowering by the time I needed it to. That was last year. This year, I bought everything.
So the next question is for Michael. Jean wants to know more about BeeSpotter. Does she have to be part of this I-Pollinate program and do this garden design to submit images of bees to the website?
Hi, thank you very much for the question. This is Michael. No, there is no requirement for BeeSpotter to be part of, to plant a research garden. I mean, it'd be great if you did both, but you can take photos of bees anywhere. And just to step back and describe BeeSpotter a little bit. BeeSpotter is a citizen science project run out of the University of Illinois, where we ask people to go and take photos of bees, honeybees and bumblebees, and come to our website and upload those photos and indicate where they took the photo, and optionally try to identify it if they can. And then we have somebody who goes through and identifies all of the photos that we receive to indicate which species of honeybee or bumblebee was in the photo. You can do that anywhere. We're actually available all through 4 states in the Midwest: Illinois, Indiana, Ohio, and Missouri. But for the purposes of I-Pollinate, we're focusing on Illinois in conjunction with research gardens. So we encourage you to consider doing a research garden, but to participate in BeeSpotter, it is not a requirement. All you need for BeeSpotter is a computer and a camera or smartphone. And you will need to create an account on BeeSpotter in order to be able to submit spottings. But that's a very quick and easy sign-up process. Does that help answer the question?
Michael, there was a follow-up question about whether or not there's a BeeSpotter app.
There is not currently a BeeSpotter app, though. That would be wonderful. It is something that would be very helpful if we did have that. So it's something we've kind of looked into and hope to have at some point soon. And I see another question in the chat that seems relevant. BeeSpotter is just for honeybees and bumblebees. I know there are a lot of other species of bees out there, but we're focusing on honeybees and bumblebees in part because of their pollinator, how much pollination they do, and also because that is what is reasonable to identify through photos. A lot of other bees tend to be smaller and very difficult to identify through photos. So we're trying to stick to what we feel confident we can actually do, even though there are tons of other bees out there. And you also don't have to indicate what plant the bees are seen on. If you can get a picture of the bee on the plant, that's great. And we can try to identify it ourselves. You can also indicate what plant it was on if you wish to, but it's not a requirement for submitting a photo to BeeSpotter.
So I know it's like a kind of an overwhelming amount of information because there are 3 different kind of like goals that we mentioned. But ultimately, our overall goal is to get citizens involved in data collection and to increase our overall understanding at the state level of how pollinators are doing. And so, but each group has slightly different needs. And you can really, in a lot of ways, choose your own adventure. Like if you want to do a research garden, awesome. If you just want to look at monarchs, awesome. If you just want to go collect bumblebee data as you go on walks around your neighborhood or in natural areas, awesome. Like if you want to like have a summer of pollinator data collection all the time, that is awesome also. Like I just, we wanted to give people options because we felt that all of these projects were extremely valuable, and depending on who you are and what your abilities were, we thought that this had a little bit of something for everyone. I know someone asked in the chat, was there a age requirement? And ultimately, one of the reasons that there isn't, because one of the reasons that we designed really short surveys was so that whether you were a small kid, maybe 5 years old, that can sit still for 3 minutes and do a count, count to 10 or 20, you could do this. If you're older and sitting for long periods of time is uncomfortable, you could also still participate. That was actually one of the ways that we just like kind of settled on the 3 minutes as a time for people to do their surveys.
Okay, trying to get through the questions. I have, I think, 4 questions for Morgan. Morgan, if you register for the webinar, are you registered for the garden?
Great question. Unfortunately, no. We will ask that you go to the I-Pollinate website and register there. That would be most appreciated.
Do you make the addresses of the gardens public?
Not at all. No, ma'am. The addresses are simply for us to see sort of the distribution of the gardens, to see where data are being collected. So no, the addresses are simply for our internal use. They are not public.
And will the data be shared after the last recording in September, and when would that likely be?
Yes, it certainly will be shared. We will put together a sort of breakdown of the preliminary results, how many surveys were conducted, what types of insects were observed, things like that, and we will send it out either through the mail or electronically. That will probably be in November, December. So we have time to go through, clean the data, analyze the data, and put together that document.
Can you describe a little bit about the scientist visits that we talk about on the website?
Yes, certainly. That is also a great question. So in the garden registration, there is a question that asks if you would be comfortable with a scientist visiting your research garden. By all means, if you are not comfortable with that, just click no. We will never contact you and ask you. We certainly won't just show up. That would be absurd. But we, the reason that we request that option to visit is simply for clarification and verification of the data. So if I come out and I visit a garden and I observe 157 bees on one plant, and someone else did their observation and they observed 100,057, you know, that's a discrepancy there, and that's a way that we can sort of check and verify that the data is being collected in the same way across the board.
Okay, these next two questions are for David. I know he's not on the call, but I'm sure Alexandra or Morgan can answer it. Do you want them to treat their plants to eliminate the aphids?
No, because we couldn't, we discussed this for a long time and we couldn't come up with a good solution that would treat the aphids and not affect the eggs and larvae and other things that might be on the plants. So no, I mean, the only thing that you could do is maybe manual removal, but which for aphids would be awful to say the least. Someone asked if they could spray them really hard with water, but that probably would, I mean, it would probably knock off some of the aphids, but it probably would also, if there were any eggs on it, also knock them off. So unfortunately, we don't have a good solution against the aphids right now.
So they were part of the I-Pollinate garden last year. Do they have to replant their swamp milkweed or continue to record data on the second year milkweed?
I really wish David was here because my understanding of the monarchs is that age actually really matters for how attractive they are to these plants. And so if you had like a 5-year-old stand of swamp milkweed, they're going to actually be less attracted to them than they are to the newer plants. And so David really encourages us to put in new plants. I definitely have seen some of my plants from last year starting to resprout, and so I'm actually thinking about moving them into a different part of my yard and not recording data on them and just using the fresh plants that I ordered for data collection this year. Second year plants might be okay. I would really need him to answer that though. I think that ideally it would be all new plants.
Yes, I think I remember him saying that too, that he wanted, you know, new young growth. So I know it makes it a little difficult from year to year, but that's kind of what he's going for. So the next question is, I'm sorry, I'm not keeping up. You're talking about accessible, okay. So the next question is, do you count on the Monarch survey, do you count all the monarchs on one entire plant?
Yes, you will count. So like you guys mentioned, these are going to be hopefully first year swamp milkweed plants, so they shouldn't be too terribly large. So you'll simply look at each leaf on each plant, each living plant. try to overlook those aphids and see if there are any eggs or larvae present. So hopefully it shouldn't be too time consuming considering the plants will be pretty small.
So that answered Patty Kale's question on can she do the Monarch survey on existing swamp milkweed. We would prefer that you do it, you know, with the newly planted milkweed. Now, this is a great question for Morgan. They're talking about timing here. What's the significance of the 3rd week of June?
There is no significance whatsoever. It's simply the third full week in June. This year is National Pollinator Week.
It's always National Pollinator Week.
Great. Okay, so there's some consistency. So 3rd full week in June, National Pollinator Week, and to make it easy, we simply made it the same week for every month. So it's easy to remember.
This is a cool question from Christina. If they have a beehive in their yard, does this make sense for them to sign up?
Hi, this is Michael again with BeeSpotter. Although that question might be for multiple projects. As far as BeeSpotter, we'd recommend maybe not submitting photos from your yard of honeybees, but bumblebees you could still submit, and you might want to go elsewhere if you wanted to get photos of honeybees, because it would be kind of skewing the results to get a bunch of submissions right next to an actual honeybee hive. And then I think that question could also apply to the research garden, so I'll hand it over to you guys for that one.
Sorry, I heard Michael say he was going to hand it over, but I'm like scrolling through the chat and reading, and so I didn't know if he's handing over.
Oh yeah, sure. Sorry, that was a question about if you have a beehive, a honeybee hive in your yard, how that would impact your participation.
Yeah, if you have a honeybee hive, that might make it tough to see a lot of other native bees and other insects and organisms. I don't want to. That's actually really quite a tough one, because I can see where we would get a lot of valuable data on the annuals. If the honeybees are attracted to them, that's really useful. I can also see it being really overwhelming to try and watch a small group of plants if you have a honeybee hive really close to that, because the number of bees coming at even during 3 minutes is probably, if it's something that's attractive, might be very high, and that might be quite difficult.
Yeah, I thought that was an interesting question too. It definitely would need to be noted that you have a beehive in the garden. So Jennifer says that she has 2 small empty raised beds that are about 4 feet apart. Would it be okay to do the I-Pollinate garden in those?
Yeah, absolutely. That would be perfect. So we are hoping that the beds are about a meter or so away from other plants, the I-Pollinate garden beds. And so if you have 2 that are a meter from one another, that's perfect. The one challenge might be, Morgan, we may need to work with you to figure out a way to record the beds separately, because right now, the way I think we're mostly identifying the beds based on their address. So if you have 2 at the same address, we may need to come up with like an A/B system to make sure that we know that this data came from this bed and the other data came from the other bed, if that makes sense.
Sure. And I just responded to Jennifer's question about the 2 garden beds. There is a place when you are inputting your data into the data submission surveys that you can enter relevant information that we might need. And this is also a good area to, if your research garden is in containers versus planted in the soil, that would be a great place to enter that information too, so we're aware of it. But specifically for Jennifer's purpose, if you would simply enter that information about the 2 separate garden beds into that field, that would be great.
Michael, there's a question on here about identification guides for bumblebees and honeybees.
Yes, there is, the BeeSpotter website has some identification keys and field guides available on the website. I just posted a link in the chat to a page that has a link to a number of those resources. You can also find them from the BeeSpotter website by clicking on topics in the main navigation menu. And we have a variety of pages or articles of information, and some of those involve keys and guides to understanding how to identify honeybees and bumblebees, which actually could be useful as well when you're trying to identify the pollinators that are coming to your plants for the research garden. So even if you're not planning to participate in BeeSpotter, I would recommend going and checking those out so that you're familiar with how to tell if what you're seeing is in fact a honeybee or not, or a bumblebee or not. I would also just like to throw out a little plug for, we have a page on mimics. There are a lot of bee mimics out there. So you may think that you're seeing a bee, but just because it's yellow and black does not mean that it is a bee. There are a lot of flies out there and other insects which try to mimic bees and do a very good job at it.
And there's a comparison on the I-Pollinate website of that, of how to distinguish between a bee and a fly. I guess this is a good question for Candace. Does swamp milkweed spread?
I have found it to spread some by seed, yeah. So if the seed is released and it drops in a particular area, it certainly can pop up in new areas. You could probably speak to that too.
I can, but I'm the moderator here.
Well, go. You can still say.
And then, if they wanted to save milkweed seeds, do you want to answer that question too?
Sure. You can certainly collect seed from any of the milkweed species in the fall. And typically what I will do is go ahead and plant that seed in the fall so that it can get that cold, wet treatment of winter so that it's ready to germinate for you in the spring. But you could also collect it and start the seed indoors as well.
And I just put my seed in the refrigerator to get that cold treatment.
That works too. Yep. Yep.
So there was a question about the color. So I know that Alexandra has, 12 species, but then she has particular colors too that you can mark off. Did you want to add to that, Alexandra?
Yeah, no, the color, we do want you to have the same color, and varietal is really hard, but color is important. We know that insects see, bees especially see more in the blues and yellows. We know that, I guess, mostly for honeybees and some bumblebees. But so controlling the color is, as best you can, is important. Although I have noticed sometimes I'm looking at a plant and I was like, I really thought that was more yellow when I bought it. So I guess just do the best that you can. Submit, just all you can do is submit what you have planted when you register your garden. So there was an excellent question, and I've been trying to scroll back and find it, about how to do the observations. And this is an area where people felt very anxious, I think, about whether or not they were doing it correctly, particularly because for the ornamental plants, we asked people to mark down whether or not it was a bee, a fly, a butterfly, I think an other. I think there are 4 categories: bee, fly, butterfly, and then I think other. Now, there are many flies that look very much like bees, and we do have some identification materials to help you with that. And there's also how to go about it, how to do it well. So I will tell you what I do. I take a bucket and I sit my bucket down next to my my bed, And I try and sit as still as I can because a lot of movement will scare things away. I pick the 2 plants. So we ask you to plant 4, but then just to look at 2 of them. Look at those 2 plants and see if I see anything coming into that observation area that I'm looking at of those 2 plants. Now, for some plants, this is really easy. You know, they don't have a lot of flowers, but for some plants, it's actually very difficult because, like, my alyssums got really huge. And, you know, I'm looking at 2 plants, but 2 plants that have like thousands of flowers on them. So that was a little bit difficult. But that's the best way that we can control it, is just to know that you were looking at those 2 plants. And then anytime something comes to those 2 plants, I just write down, you know, 1 bee. I usually use tally marks, so I'm keeping track of, like, that's a bee, this is a fly, a butterfly came. Which is one of the reasons that we wanted to try and have scientists also do some of the collections at people's homes, just to try and back calculate how similar is the data from that the scientist that knows what a bee, a fly, butterfly looks like is compared to what you're identifying. I don't expect you to do perfectly. It would be really difficult to, but I think you can do I think actually you'd be surprised at how well you can do once you get really comfortable with looking at things that are flies. And especially, like, what is a fly? Like, those can be pretty difficult. They can be difficult, but once you know what you're looking at, like, I was able to teach my husband really easily, and now he really can very clearly look at something and say, like, oh, that's definitely a fly, and that's definitely a bee. Once I explained to him, like, kind of like some of the like subtle differences.
Just to jump in, Alex, on the Pollinator Survey data collection sheet that I've got on the screen here, you can see where it says bees, butterflies, moths, flies, and other. There's a little bit of a description below each of the headings of those different insect types that gives you kind of an indication of what to look for when you're identifying them. So, for instance, if you're trying to differentiate between bees and flies, bees have dramatically longer antennae. They have 4 wings instead of 2 wings, and they have 3 distinct body segments, as opposed to flies that have just 2 wings and 2 body segments. So if you're feeling a little bit uncomfortable, that can be hopefully a resource for you.
And their eyes cover their whole face, unlike the bees. That's one of my key things of distinguishing them out.
Right, good point, Kelly.
I know this is not a question, but Melody writes, I participated last year and it was fun. It was a lot of work to learn at first, as I'm a novice gardener, but was less time as the project went on. This is the first time in my life I ever sat down and looked at flowers for 3 minutes straight at a time. I encourage everyone to at least try it. I definitely felt the same way as Melody. Like, you look at plants and you look at flowers, but to really look at them, this project really gets you to really look at flowers and the insects that are visiting them. So I really thought that comment was cool. I felt the same way as Melody. And then team, am I missing any questions? I see there are 4 new.
There was one so someone asked that they did it previously, and they said one of the challenges that they had was knowing whether or not they should count an insect that moves onto your plant as being the same or different. And and this is actually something I realized we did not clarify in the recordings. If it moves outside of the 2 plants that you're looking at and comes back, I count it as new. If it stays on those 2 plants, I just count it as 1 visit. And that can be really hard, and you're probably not gonna, it's not gonna be perfect, but I think just knowing that when something moves into your plant, onto the 2 plants that you're watching, count that as the visit. And then, like I said, if they stay on those plants or walk around them or move to the other plant that you're looking at, just count that as 1. And if it moves out of your those 2 plants and then comes back, because once something comes in, it's hard to know for certain if it's new or old. or something that you've counted previously. So that's how I make my distinction about whether or not I'm counting something. Like if it's a new visitor onto the 2 plants that I'm looking at, count that as 1 visit.
I know, Alexander, several times I would be doing my 3-minute observations and there'd be a butterfly flying around and I'd be like, land, land, land. And it never would land, so I couldn't count it. I did forget one question, the black walnut question. Unfortunately, I am going to have to ask Candace about this. I don't know if we know exactly what plants can grow under a black walnut and what plants can't. I'm sure there's some lists out there. Candace, can you add anything?
Yeah, I don't know off the top of my head either. Let me do a quick Google search. I'm sure there's some good extension charts of what is and what isn't. So let me see if I can find one quick and I'll link it.
Yeah, that might be a good situation where container gardens might work out really well too. Like rather than putting them into the ground.
Yep, for sure.
Someone asked if they weed. Yeah, absolutely. I mean, you don't have to, but I found that if I didn't at least do something, there seemed to the henbit really crept in pretty hard into my garden.
So they should just do their normal general gardening practices for plant health, correct?
Yes.
I just linked Wisconsin and Purdue fact sheet has some charts on it, so you can check that.
Thank you. Are we done with questions? I know I'm supposed to be moderating, but man, they came in so fast.
Yeah, I really appreciate people hanging with us. I mean, I think at a peak we had about 115 participants, and that's amazing. I would love to have 115 people reporting data. I cannot thank you enough for your interest and willingness to participate in this project. And if you have questions, you can, there is a question form on the I-Pollinate website, and Morgan gets those. And she's really great at responding. So if you think of anything, please feel free to message us. And I really appreciate your participation today. Thank you.
Sure. And if I personally don't know the answer, I can always forward your question on to someone who will know, and then they can share that answer with both of us. And just like Dr. Harmon-Threet said, thank you guys so much for participating in this webinar. You guys had great questions. This was really a pleasure.
Yes, thanks everybody for hopping on. This has been recorded, so I will get this recording uploaded, and I'll send an email out to everybody that registered for the webinar. I know I did have a problem with the password on one of the emails, so I'm sorry some people weren't able to to get in today, but I've let them know that the recording will be up as soon as I can. So I'll send out an email. Thanks everybody for joining today. We appreciate it.
Thank you.
Yeah, thank you, everyone.